Good evening, all of you. Uh, I will start the session in 1 minute. Later, the students have to join. We will start in 1 minute. So, good evening, everyone. I hope you guys are fine and studying well, practicing the last assignments. I myself, Santanu Singh, PMRF scholar from IIT Varanasi. Wait a minute. So, I hope my slide is visible and audible to you. Uh, so, Myself, Santanu Singh, PhD scholar from IIT Varanasi, PMRF scholar from Cycle 9 with PMRF ID 1102015. Today, today is the week 3 of the problem solving assignment. Uh, so, in today's session, we will solve the problems of week 0 and week 1. We will try to just uh, elaborate or explain those questions which are you unable to solve, and if you have any queries between them you can directly ask to me. So before starting, I will brief about the course outline. Yes, this course name is Demystifying the Brain, and the course code is BT17. Course ID is NOC BT17. This course is of duration of 4 weeks, and the course instructor is Professor Srinivas Chakravarti from IIT Madras. I am assigned as the PMRF TA for this course, and I am from the IIT BHU Varanasi. This course comes under the Biological Sciences and Bioengineering under the SWAM online free education courses. So the course layout is having, let me admit other students also. Yeah, okay. So guys, please be on time, and we will have a good session today. So I was briefing the course layout. Yes, this course having the week 1, we will have a study about the history of neuroscience, brain through evolution, which is completed by the course instructor. And week 2, we will study about the neurons and neuron signaling. And week 3, organization of the nervous system and maps in the brain. The last week will be completed by after 16 February, that is on the memories and holograms, emotions in the brain and theory consciousness. I recommend you guys to study these two books, Phantoms in the Brain and Demystifying the Brain. I personally recommend you this Demystifying the Brain. It's easy to read and on a simple language with a free access on the NPTEL website, which was written by Professor V.S. Chakravarti. So in today's session, what we will perform, yes, the problem solving session of the assignments of week 0. Uh, in between the session, if you have any doubts, please unmute yourself and or either type your doubts on the chat box. By any chance, if you not able to ask any doubts, you can directly email to me on this my email. I will reply with you with the possible answers. Okay. So every week timings are 3 p.m. to 5 p.m. By chance, if you miss those classes, you can directly find the recorded video lectures, which are available at both NPTEL website here, SWAM courses, and on the YouTube channel. Okay. So let's start the today's session. So in week 1, assignment 1, you have been asked about the which philosopher scientist suggested that the function of the brain is to cool and the pumped by the heart. So this statement is wrong. But in ancient times, when we don't have any facilities, labs, do not have any infrastructures, so the scientists have predicted that the function of the brain is to cool the heart by pumping the blood. But now it is just like reciprocal. Okay, heart pumps the blood to the brain and it's cool. So in this question, you have been asked that which philosopher or either scientist have suggested these questions. So if you know the answer, you can directly write the answers in your chat box. If you don't know the answer, don't worry, I will let you know. So guys, are you trying? Okay. So here is the interface of the NPTEL website. By chance, if you miss, you can go through it. Assignments about the course, progress, mentor, and you can also review your assignments. Okay. So back to the session. So I hope you guys been trying the answers. So let's solve this question. The correct answer is Aristotle. We have the 4 options: Robert Hooke, Aristotle, Santiago Ramon C. Segel, and Ehrlichman. So the correct answer is Aristotle. Why? In ancient, medical practitioner has conflicting views of the significance of the brain. Okay, that's right. In the 4th century BC, Aristotle considered the brain to be a secondary organ. Okay, what is primary organ, what is secondary organ. When we give the importance to organ that will control the body, we will consider it as a primary organ, and we don't and when those organs will not consider as the controlling or mastermind of the body, we will consider as the secondary organs. So what would be other secondary organs of the body, just like kidney, liver, and all? Primary organ is brain, heart. Okay, so Aristotle considered the brain to be a secondary organ. That was not true right now. That served as a cooling agent for the heart. Okay, and a place in which spirit circulated freely. He designated the space in which all the spirits came together as the senses. So at that time, Aristotle was a philosopher, because due to the lack of the instruments, he given a theory that uh, brain pumps the blood to the heart and it is a cooling organ. But it was wrong, and it was just vice versa. Now, with the help of advanced techniques such as EEG, PET, positron emission tomography, uh, electron encephalography, and other imaging techniques, we will directly know that yes, cerebellum circulation is responsible for the providing nutrients to the brain, and heart is only the pumping organ which pumps the blood to the brain and other organs of the body. Okay, so movement of the blood through the vessels that supply the brain and surrounding structures. Okay, Aristotle had famously written, there is nothing in the intellect and that is not in the senses. So I hope this question is clear to you. Let me repeat the question with the correct answer. Which philosopher oblique scientist suggested that the function of the brain is to cool the blood pumped by the heart? So the correct answer is Aristotle, and here I had explained about it. You will find the PowerPoint link at the NPTEL website. You can directly go through it, the completed assignments, and you can download this PPT. I will upload that on the website. You can directly go and download it. Okay, by chance if you missed my class. So moving to the next question. Uh, the second question which you have been asked, that which lobe was wider than the normal in Einstein's brain. Okay, actually just here is the typological error in an Einstein having the one space. Don't mind that. So we have the five options: frontal lobe, inferior parietal lobe, superior parietal lobe, temporal lobe, and occipital lobe. So guys, Einstein is a great scientist. Okay, when he died, the scientists had isolated their brain, and they performed several studies on his brain. And what they found, that the Einstein brain was not normal, it having some different structures than the normal brain. What are those structures? We will let you know. I will let you know. And if you have attended the lecture of week 1, uh, hope you can able to answer this question. Okay. So in 30 seconds I will give you the correct answer with the proper explanation. But before, you can try to the giving correct answers by writing the correct answer in the chat box. Okay. So which lobe was wider than the normal in Einstein's brain? Okay. The correct answer is inferior parietal lobe. Why? Parietal lobes are responsible for the visual and 3D representation and mathematical reasoning. Is inferior parietal lobes are not divided by the major cleft. Not seen in 191 controls, axons were connected in unusual ways. Might have allowed for his brilliance and his ability to put the spatial representation into mathematical concepts. Einstein was a great mathematician and physician. So he do the calculation very easily, which we cannot able to do. So you can see a typical brain in the upper left image, Selvian fissure and parietal operculum having the different structures. But in Einstein brain, parietal operculum you can see cannot found, and the parietal lobe, inferior parietal lobe is bigger. Okay, bigger or say wider. So Einstein, due to this advantage, Einstein was able to solve the questions very easy and solve the mathematics problem very easy. The synaptic transmission is very fast, and he is able to solve the mathematics problems very easily. Okay, so let's move to the another question. What is safe wire design philosophy of the brain? What is safe wire design philosophy of the brain? We have the four options again. The tendency to minimize the total wire length. Second option, reduce the total conduction delay by myelinating axon. Third option, the connection between two neurons gets strengthened if they are simultaneously active. Fourth option, saving the wire, dendrites and axons at the expense of somas. I hope you guys attended the lecture and you will able to solve. I'll give you 30 seconds to answer this question. Guys, please try. If you will try, I will explain you better. If you don't try, might not, I will explain better. Okay, so guys, please try. It's upon you. Okay, so we are moving to the correct answer. The correct answer is tendency to minimize the total wire length. Okay, let me repeat the questions once again. What is safe wire design philosophy of the brain? What is safe wire design philosophy? Okay, a range of neuroanatomical results supports the idea that safe wire is an organizing principle of the brain structure, that the theory of the combinational optimization of the networks applies to the anatomy of the nervous system. You know, it's the basic structure of the brain, having the neuron connections which we'll consider as the total wire length, and the tendency to minimize the total wire length is the safe wire principle, in which in particular optimization of the placement of components operates at several hierarchical levels in the nervous system, from the gross microscopy anatomy and from the invertebrates to primates. Okay, got it? That is when anatomical positioning of the interconnected neuron components is treated like a problem of wire minimization. In microchip layout, a hypothesis of the best of possible brains is consistent with the observed sitting of brains, ganglia and even somata of the individual neurons that minimizes the length of the sorry interconnections. In case of the positioning of ganglia of the cannabis elegans, C. elegans, optimization prediction of 1 in a million precision can be verified. Okay, so it is the explanation of the theory that same wire design philosophy of the brain. Okay, so the correct answer is tendency to minimize the total wire length. The incorrect answer is reduce the total conduction delay by myelinating axon, and the connection between two neurons gets strengthened if they are simultaneously active. These all three options are incorrect. The correct option is the tendency to minimize the total wire length. Okay, here I had explained very briefly about that, why this option is correct. If you have any doubts, you can email to me or you can write in the chat box. I will able to answer you. Okay, so the next question which was asked on the week assignment 1, the question 4 is that multiple sclerosis is caused due to. We have again four options: demyelination of axon. Second option, selective death of the dopaminergic cells in substantia nigra. Third options, degeneration of the glial cells of the central nervous system, and all of the above. Guys, you know, when I was taking the week 1 lecture, I had explained very briefly about the multiple sclerosis. Okay, so I am damn sure that you guys will know the correct answer. If you don't know the correct answer, no worries, I will explain. If you know the correct answer, please answer it and write in the chat box. I'll give you 30 seconds time. Okay. Okay, guys. So I hope you have uh, tried to solve the questions. So what the question is, multiple sclerosis disease is caused due to. Okay, first let me know. Multiple sclerosis is the disease which in which the demyelination of the axon occurs. Okay, so it is a neurodegenerative disease which occur in the axons where the demyelination occurs. Okay, so here is the correct explanation. You can see in the in the figure. Here is the normal neuron, which having the axon, myelin, and nucleus, and here is the demyelinated axon. So in the multiple sclerosis, what happened? You can see the normal axon. It is polished and uh, continued, but in case of multiple sclerosis, it is damaged. Okay, so when the axon become damaged, what will happen? The nerve transmission is affected. So the, that transmission will cause the cognition, neurological problems, and a disease called multiple sclerosis. Okay, so the explanation is the myelin sheath is a fatty covering around nerve fibers. Okay, this is the myelin sheath. It is a fatty covering around the nerve fiber. This is the nerve fiber. Okay, so that helps them to send the electrical signals. Yes, of course, we had studied about the action potential, depolarization, hyperpolarization, and repolarization. So these all are the electrical signals which are very essential for the neuron transmission. Okay, so demyelination refers to the destruction and loss of this sheath. Okay, which leads to the development of lesions where the tissue is damaged and scarred. A lesion is also maybe called a scar or a plaque. Okay, so it is the when the scar or plaque is formed, it is the worst case of or chronic case of multiple sclerosis. In initial, what will happen? The fatty covering of the axon will removed. Okay, I hope you guys are able to solve this question. If you have any doubt about the multiple sclerosis, you can ask. There are several other diseases just like Alzheimer's, Parkinson's. These all are also the disease related to neurodegeneration and the brain. Okay, I, rec- I recommend all of you that please also go through these diseases and study very briefly that what is Alzheimer's disease, what is Parkinson disease, what are the treatments for multiple sclerosis, what are the treatments for Alzheimer's, what are treatments for Parkinson's. You know, the that will help you to study more interest. That will make your study more interesting. Okay, so coming back to the topic, for this question, multiple sclerosis is a disease caused due to the correct option is demyelination of the axon. Okay, why? Because the fatty covering around the nerve fiber will removed or damaged. Okay, so there will be a demyelination in the multiple sclerosis. I hope you guys having uh, no doubts in this uh, question. If you are not able to understand this question, please raise your hands. Okay, are you guys understanding? Yeah. Okay. So I have seen messages that yes, this concept is clear. So guys, when we are solving one question, it is not like that you have just revised those questions and uh, it is completed. I recommend you to please go through the another questions also and another topics also. Okay, just like if I told you the multiple sclerosis pathophysiology, you can please go through the treatment, symptoms, and other factors which affect the multiple sclerosis. Okay, so moving to the next question. This is the question number 5. The four components of neuron signalings are arranged in the correct order. This question has been solved already in problem solving assignment week 1. So let's repeat this question because it is repeated on your assignments. The four components of the neuron signaling are arranged in the correct order. So you have to arrange this sequence in a correct order. The signal transmission from one neuron to another neuron. The first option is action potential generation in the cell body, to the signal propagation along dendrites, to the signal propagation along axon, and signaling across synapse. Guys, it is the correct answer. Do you know about it? No? Okay, let's try another option. Signal propagation along dendrites, signal propagation along axon, signaling across synapse, action potential generated in cell body. The option 3 is signal propagation along dendrites, action potential generation in cell body, signal propagation along axon, and signaling across synapse, or either the fourth option, none of the above. So in the lecture, there is a very clear-cut solution of this question. If you try to think, you will able to answer it very clearly and very correctly. So I give you 30 seconds time. Please comment your answers as A, B, C, D. Okay. If you don't know about it, don't worry. I will solve this question just after 30 seconds. Okay. Try to solve, guys. Okay, I hope you guys have tried. Please write your answers in the chat box so that I'll able to understand that you guys are able to solve or not. Okay, the correct answer is signal propagation along dendrites, action potential generation in cell body, and signal propagation along axon and signaling across synapse. So here is the question. Four components. There are four components. What are those four components? Dendrites, action potential generation in the cell body, then signal propagation along axon, and signaling along synapse. So here is your dendrites. You can see my cursor. These here all present are the dendrites. Then action potential generates at cell body. If you attended my lecture 1, I had explained very uh, thoroughly about the action potential and all things. So action potential is generated on the cell body. I already explained that. And then axon and then synapse. Here you can see the zoomed structure of the synapse. presynaptic membrane, postsynaptic membrane, synaptic vesicles, neurotransmitter release, receptors which captures those neurotransmitters, and this space is called the synaptic cleft. Okay, so if you have any doubt about this explanation, please raise your hands or either ask your questions. If not, we can proceed further. So I assume that you guys have no doubts and I proceed further. Uttam, do you have any doubt? Yeah, okay. So the neurotransmitter is nothing but a signal which releases from the synaptic vesicle and binds to the receptor and transfer the signal from one nerve to the another nerve. Okay. Neurotransmitter may be acetylcholine, dopamine, epinephrine, norepinephrine. These all are the neurotransmitters. Okay. So we'll move to the next question. Question number 6. What consists of the recording of the voltage produced by the brain's electrical activity by placing electrodes on the scalp? This was first recorded by which scientist? Okay, so guys, this question I already explained, which was repeated in your assignment. Please answer. If you not able to answer, I will explain, don't worry. So okay guys, let's try to solve this question. What consists of the recording of the voltage produced by the brains? This question is very clear that the recording of the voltage produced by the brains was only done by EEG, electroencephalogram, okay, by placing the electrodes on the scalp. So this might be the correct option, the first recorded by the Hansberger. Let's see. Yes, I was right. EEG Hansberger, okay. So EEG consists of the recording of the voltage produced by the brain's electrical activity by placing electrodes on the scalp. Okay, this was first recorded by the Hansberger. Why? Let's see. An electroencephalogram (EEG) is recording of the normally occurring brain electrical activity. The recording is made by placing a small metal disc called electrodes on the patient's scalp. The electrodes pick up the electrical signals produced by their brain. Okay, so as you can see, this is the here is the patient which head is covered by the electrode, and this is the EEG cap, and there are several electrodes which captures the signals, and it is connected with the amplifier. The purpose of the amplifier is to amplify the signals, because the electrodes which capture the signals are very less. We need to amplify those signals. When the signal is amplified, it is measured in a graph. You can see measured potential for each electrode, this kind of graph, and it is processing. So what will happen in the processing? We can easily differentiate between the normal and disease condition. The normal condition will show different graph. For the electrical signals and disease condition will have the different graphs. Okay, so we can directly compare to it. Okay, so the correct option is EEG consists of electrical recording of the voltage produced by the brain electrical activity by placing electrodes on the scalp, and it was first recorded by Hansberger. Okay, here is the explanation. So we will move to the next lecture, next question. Here is the question number 7. In case of feed forward connections, projection from a large brain area to a small brain area is said to be blank, whereas a projection in opposite direction is said to be, if you are able to recall, in the second or third lecture uh, of the Professor Srinivas Chakrabarty, he has clearly told about the convergent and divergent connections. So those students which are able to recall, they can answer the question. If you don't able to recall, I will explain. Okay, I will give you so this question is slightly tricky, so I will give you 1 minute to solve this question. Okay, so okay guys, we will try to solve this question. So in case of feed forward connections, a projection from the large brain area to a small brain area is said to be, whereas the projection in the opposite direction is said to be. So there will be convergent and divergent, but you guys have to think. So the correct option is convergent and divergent. So here it will be convergent. The option 2 is correct. In case of feed forward connection or projection from large brain area to small brain area is said to be convergent. Okay, whereas a projection in the opposite direction is said to be divergent. Okay. Why? You can see convergence and divergence. When a presynaptic inputs, convergence of the input, one cell is influenced by others. It goes to the postsynaptic neuron and then presynaptic inputs. So when the signal comes from different uh, other cells to one neuron, it is called as convergence signals, and the signals distributed to various other neurons, it is called divergence. Okay, so be specifically and clear about the convergence and divergence. Always convergence means the 3 to 5 signals are coming from different sources and coming to one, and divergence means dividing, it is the one signal is divided into many parts, so it is called as convergence, and here left right one is called divergence. Okay, you see the figure, you will clearly try to solve. So okay, I will give you 10 minutes to solve these questions with a proper explanation. Okay, okay, let me remove the answers. Now you guys can answer. I'll give you 10 minutes to solve this all three questions. First question is, which of the following is an accurate instance of loop of connection in the brain? Here are the options. And the second question is, which of the following accurately describe the sequence of evolutionary stages of neuroanatomy? The next question is, which of the following electrical response in a neuron is said to be all or none response? Okay, so guys, you have 10 minutes to solve this question. If you are not able to solve, I will explain. Okay, but first try to solve this question. Okay, you have the 10 minutes. So guys, I hope you guys have solved these questions. If not, let's try to solve this question. So these are the correct options. Which of the following is an accurate instant loop of connections in the brain? The correct option is corticosome fibers, neostriatum, globus pallidum external and globus pallidum internal, thalamus, corticosome fibers. The next question is which of the following accurately describe the sequence of evolutionary stages of neuroanatomy? Okay. The correct option is diffuse nerve net, chains of ganglia, small brain, large brain spinal cord, large brain, small brain spinal cord. Okay. And the next question is next and final question is which of the following electrical response in a neuron is said to be all or none response? I in my first lecture I had told about the all and none response. So it is very obvious that the correct option is action potential. We have a detailed discussion of action potential on the lecture 2. Please go through it, and those who have answered the correct, congratulations to those of them. They are still learning and trying to get the correct answers, and those who have given the wrong answer, please don't worry, do study, keep revising. Okay, so again, if you have any doubts, you can directly email to me on this email, santhanusingh.rsbc21@itbhu.ac.in. It is my official email. If you by chance if you have any queries, you can directly email to me. Okay, so thank you so much, and keep learning, don't lose learning. That will help you to achieve your goals. Okay, thank you so much. Now you guys can leave the class. Those having the doubts, they can just unmute yourself and ask the questions. If you don't have the doubts, you can leave the lecture, and if you have any doubts after this class, you can email to me or you can write in the chat box. Okay, so thank you so much guys. Please, I request all of you to please come on time on 3 p.m. so that we have a good discussion. Okay, on the next week. Thank you so much. Thank you so much. Yeah, please left the class. If you have completed, please left the class. Okay. Yeah, okay. Thank you.